I'm going to talk to you a little bit about the work I've been doing for my current postdoc at the University of Missouri St. Louis. So as biologists, revolutionary biologists, we usually start with a good and interesting question, or at least that's what we try, and then very quickly start thinking about the data that we are going to generate, or what we're going to get to answer that question. And so in this great genomics era that we're in, there are many ways to get molecular data. And so you have things like microfluidic PCR, transcriptomics, whole genome sequencing. But I'm going to talk to you a little bit about the three ways we're using in this project, which are shotgun sequencing, sequence capture, and good old Sanger sequencing. So I've been working on the plant genus Burmeistera. It's in Campanulaceae, or the bellflower family. It was described in 1854. There's around 116 species distributed from Guatemala to Peru. These are shrubs that grow on understories of dog forests. And the highest species richness is in Colombia and Ecuador, where there's around 50 species of them. In 1943, there's this taxonomic treatment of the genus, where Wimmer described two different morphological sections based on the presence or absence of hairs on the anthers. Burmeistera usually has these green flowers that are pollinated by nectar-feeding bats in the family Phylogenomics. Here are two examples of pollination in action by species of the genus Andean. And my postdoc advisor has done a really good job in studying some of these pollination syndromes in a few species of Burmeistera, and has shown that there's a strong mechanistic isolation based on the exertion of the reproductive flowers, of the parts of the flower, which is the distance between the tip of the anther to a constriction of the corolla tube. And it's important because there are species that have very short exertions and some that have very long exertions. And so this results in very specific pollen placement on the head of the bat that pollinates them. He's also shown that there's a strong relationship between the length of the corolla tube with the tongue of the bat that pollinates them. And some of these bats have really long tongues, almost 3 inches long. Burmeistera has been included in a few broad-scale analysis. My lab mate Laura Lagomarsino has shown that the genus is monophyletic, that it's closely related to the genera Centropogon and Cyphocampulus. Recently, she also showed that the median crown age of the group is 2.6 million years, and that there are very interesting diversification dynamics in this clade based on the fruit type you have or the pollinator that visits you. So I joined the lab, and there were all these studies that had used some or a few species of Burmeistera, and so there were all these data just waiting for someone to get them. And also, luckily, they had just sequenced 19 species using shotgun libraries, and they had assembled whole plastons for all of them. So sort of my first task was to find a way to combine these two data sets, Sanger and whole plastons. So I went to GenBank, I got around 200 accessions and 7 chloroplast loci, these 19 plastons. And so the first way I tried to put these things together is what I'm calling the everything approach, and that's literally just putting everything in a single alignment. This matrix was comprised of 50 species, maximize the amount of data that I had, but also the amount of missing data that I had. And so, because these 7 loci are only around 7% of the plaston, and we know that a lot of missing data can adversely impact your phylogenetic inference, and when we build a tree, that's exactly what we get. We get this weird-looking clade here, and it's comprised of species that have the least amount of data. And also the support is super low, and so we know that this tree is no good. And so the second approach I tried with something I'm calling extraction. I start with the same matrix, but now I get rid of the regions that are not shared between the Sanger data set and the high-throughput sequencing data. And then this results in a much cleaner data set with a lot less missing data. We build a tree, we see that the missing data clade is gone, that the support is a lot higher, but we're also throwing away a lot of the data from the whole platform. So we're like, oh, is there a better way to use this high-throughput data? And so we tried something else. We started with the plastons by themselves, aligned them, partitioned them by coding and non-coding regions, run this through Partition Finder to find the best partitioning scheme for those data, and then ran a partition analysis. This gave us a complete plastron tree for 19 species, very highly supported, that we then use as a constraint for the extracted data set. So basically, we're just letting these species that have only standard data to fall wherever they belong on the tree. This gave us a very similar tree, supports a little higher, and I think we didn't see that much increase in support because we only have 50 species. This method has been used in larger clades, for example, acacia with 500 species, and they've shown that having a constraint tree can really impact the results. But regardless, we were able to make some biological inferences. For example, we see that there's some geographic patterns. We also see that those two morphological sections I mentioned at the beginning are not monophyletic. So we started to think of possible characters that we can use for a future subgeneric classification. But we know of the limitations of the data that we had. There's only 50 species in this tree. It's a single locus phylogeny because there's no recombination in the plastron. And so we can't get at the coalescent processes that are probably playing a big role in this group. And so we decided to do sequence capture. However, the price you pay to work in this beautiful cloud forest in South America with non-model organisms is that there aren't any readily available kits that you can just order. So you have to design your own baits. And so I did this using those shotgun libraries that I had, 4 transcriptomes, a plastome and a mitogenome reference. I put all of this into this pipeline, Sondovac, which was published last year, which through all of these many steps of aligning, cleaning, assembling, mapping, gives you a list of genes that you may be able to use. And so for us, that was a list of 745 single copy nuclear genes, greater than 600 base pairs. Some of those genes have multiple exons, so we're actually targeting over 2,800 loci. And then very recently, we did a pilot run for these genes with 73 species with paired end data. And then we ran it through this other pipeline called HiPiper, which is made to deal with this kind of data. First, it takes your reads, it sorts them, and bins them based on the target they come from, then assembles those reads de novo, takes those contexts and aligns them to the reference they belong to, and then if there's multiple exons, it separates them, it tries to assemble flanking regions. And so you end up with some intronic sequences that are going to be likely more informative. Our experiment worked really well. As you can see here, these are samples by loci, the darker the better. On average, 85% of our reads map to a target gene, which means that our bait design was very successful. And we were able to get over 500 genes for most of the species. This was very encouraging because we were using a lot of herbarium specimens, and so very good. And so plants have between 50 and 200 copies of the chloroplast in each cell. And so regardless of how much you clean the DNA and how specific your baits are, you always end up with some plastome data. And so we use this upcoming pipeline called FastPlast to generate plastome data, and we were able to get between 10 and 150,000 base pairs of plastome, which is great because this is just free data. Of course, we built gene trees for all this, and not surprisingly, those gene trees are not very good. In blue, you see the outgroups, and so they're all nested within Burmeistera. But we weren't too concerned about this. Once we put this through a species tree analysis, be it SDD4 text with 4.2 million base pairs, or through Astro2 with 600 gene trees, we get that Burmeistera is monophyletic. We get very good support for the relationships of the species, and so that we were very happy about that. But this is very preliminary. We got this data a month and a half ago. And where we're really going is we want to answer what's driving speciation in Burmeistera. And specifically, we are asking one of the main questions in pollination biology. And so it's believed that usually pollinators are driving speciation, but we think that, at least in Burmeistera, is the opposite way, where there's speciation happening, and once the species have diverged, then they have character displacement to avoid competition for the pollinators. But I won't be able to show you anything about this, but hopefully very soon. And with this, I would like to thank my collaborators, funding, and I'll take any questions.